So those other slides that we just covered are in the updated slide set. But last time, I think we were talking about this 1983 study that really kind of rocked the world of a lot of plant biologists, and they got so much pushback saying that this was ridiculous, this doesn't happen, you screwed up your experimental design, you did it wrong, you analyzed your data incorrectly. And so they had to go back and do a more careful study in the lab. And in that way, they were able to show pretty much the exact same thing, that there were volatiles being given off by the tree that was getting attacked. In the field study, they actually did it with caterpillars chewing on the leaves. In the lab, they just physically tore. By the way, did you know that's a good way to increase the nutrient profile of like your leafy greens before, like when you make a salad, before you eat it, you should tear up the spinach or whatever, arugula, tear those leaves up at least a little, and then let it sit for about 15 minutes. And so, I mean, remember, those leaves are living, as long as they're not totally wilty and slimy. They're living leaves when you go and buy them. And so when you tear them, those leaves think they're getting attacked. And so they'll start ramping up their secondary metabolite production, which is healthy for us. And so it's something that I remember reading that somewhere, and I'm like, duh, of course, that I just hadn't pieced that. I'm like, oh, that makes so much sense. So anyways, they tore some leaves, and in the lab study, and basically they were showing the same thing, that the volatiles from the damaged plant could impact any of the leaves or plants nearby. But if you had another plant that was totally encased, where the gases, the atmospheric gases couldn't get to that plant, then they didn't exhibit any change in their behavior. And so we finished that, and then I wanted to continue. So this raises the idea that, okay, if plants are giving off volatiles when they're under attack, are they doing that on purpose to warn other plants? And so there's a whole field of study where they get into this idea. Is it altruistic? Like they're warning others, hey, I'm getting attacked, you're next? Is it just a matter of plants nearby happen to hear the signal, they're eavesdropping, and the plant that is releasing the volatiles really isn't trying to do anything with that? And so this has been a big, again, debate. So when a plant releases a smell in the air, is it a form of talking, or is it, so to say, just passing gas? And so while the idea of calling out for help or warning its neighbors is fun to think about, is it really the intent? And so this is another area that is no decision is being like firmly said about it, but we will come back to this idea when we look at plant-plant communication in more depth. Thinking about this idea, it's challenging to design an experiment to get at these ideas, but they're trying very hard. One early experiment that was being done, just again to kind of show that the volatiles definitely are playing a role. This was a different experiment where these were lima bean plants, and they put beetles onto one of the leaves. And so that's supposed to be a beetle on the very left there, right here, sitting on a leaf. Okay. And then they had a nearby plant. So that's the one in the middle where they shared air with the first plant that had the beetle on it. Then the third plant, or the third set of plants, I colored that yellow to try to show that it was encased, so that any volatiles coming from that first plant could not get to it. All right. So then they released the beetle, let it chew, and they took the leaf that was chewed, so that's why I have a circle around it, and then they took a leaf from the same plant that hadn't been chewed on, but same plant, And then they took leaves from the neighboring plant and they took leaves from the encased plant. And when they were looking at volatile production, they could definitely show that the plant that had the beetle on it, not only were the leaves ramping up their defenses, both on the chewed leaf and the non-chewed leaf. And so basically that's saying that you had a systemic response. The plant was able to kind of tell the rest of its body, so systemic, to go ahead and gear up. The neighbor plant that shared the same atmosphere also had defense chemicals. So that's getting at the idea of those volatile organic compounds. I say VOCs. I've heard other people say VOX, so whichever you're used to. They also had defense chemicals ramped up in their leaves to that beetle. And then the isolated plant that was encased separately away had nothing going on, no defense chemicals. And so this was just a really nice experiment, again, showing that you can get this kind of communication going on. They did another set of experiments where they put a plastic bag around where they put the beetle on the leaf. And then had the beetle on that leaf, and that leaf still had the defense chemicals, but the other leaves on the same plant, the neighboring plant and the encased plant, had nothing. And so this was quite nicely showing that, okay, VOCs are definitely involved in this because they covered up, they put a plastic bag around the leaf. And so they captured all the VOCs. The VOCs couldn't get to the other plants. And so that second part of the experiment was really nice because it showed that, yes, there could be signaling going on through the plant body, especially with the plant that had the beetle on it. Certainly there was chemicals probably flowing through and getting to the other leaves through the plant body, but they also showed a volatile going out. And so this is actually something that we show in Plant Fizz, where they're showing salicylic acid and methyl salicylate, which are, the methyl salicylate is the volatile version of salicylic acid. And so what happens is the plant can make, depending on what's attacking them, they'll make different hormones. You don't necessarily have to memorize exactly which hormones are from which attack, but basically we have this idea where depending on if it's an herbivore chewing on a leaf or if it's a microbe that's attacking through more of a chemical attack, you'll get different hormones being expressed. So that's the way for the plant to figure out and behave appropriately, because they have to behave differently if there's a beetle versus fungus. And so here they're showing that you can have this systemic flow. So the red arrows going up the stem from the attacked area. So this is looks like they tried to put like a fungus on there. And then you can see the systemic flow through the stem to the other leaves. And then the methyl salicylate, you can see they did a squiggly line. They're up off the leaf. They're trying to show that that's a volatile. And so it changes from salicylic acid in the plant body, and then it gets volatilized to methyl salicylate and goes out of the plant body. And then nearby plants can get access to that signal from the volatile release. And so this neighboring plant will then ramp up its gene expression pattern to resist attack by that same fungus in this case. So this is something that, again, when this was first being teased apart, the idea that there's volatiles being given off was a big surprise. And they started looking at this idea more carefully, and there's some experiments that have been done that really nicely show an added twist to this idea, where in this case you have a caterpillar chewing on a leaf and the plant can feel the caterpillar. It can taste the caterpillar because as the caterpillar is chewing, the chemicals from the saliva gets into the plant. And so the plant goes, caterpillar saliva, bleh, and starts to kick in its defense system, including jasmonate or jasmonic acid, which is a hormone that the plant makes. And in this case, so the velocitin here, by the way, that's in the saliva of the caterpillar. So when the plant can detect that saliva and the chemicals, it ramps up its defense system through jasmonate production. That's part of the process. And then it actually creates a situation where it releases volatiles into the air to attract the predator of that caterpillar. And so you have the, what is it? The enemy of my enemy is my friend idea going on here. And so again, just mind-blowing experiments when they had no idea that plants were this sophisticated, and very careful studies are showing that this is accurate. Time and time again, you can see the parasitoid wasps zooming in on the caterpillar on the plant and attacking it and killing it and eating it. And so this idea that plants can have signals going through their whole body to warn other parts of their body. And now they can do volatiles and it seems like other plants are picking up on that so that they can ramp up their defenses. And then this case where it's actually volatiles that are calling in predators to get rid of the caterpillars. So please watch this. You're going to see a little bit more of that in that documentary of what plants talk about, because they get into this a little bit. Any questions on this idea? Yes.
Making jasmine to try to attract the wasps, but this recognizing that jasmonate's there and that probably means.
Good question. Yeah. I mean, jasmonic acid can be made for several reasons by the plant. And so I think what's going on in this particular case is that the jasmonic acid is part of the early signal transduction process for this happening. And then a very particular volatile organic compound gets released that the wasp, it's very particular for that wasp. But you're right, jasmonic acid can be used for a lot of reasons by the plant. It's not just for this. But the picture on the right, by the way, is just there to drive home the point. When they say vascular, that's, going through the whole body. Vascular tissue is the xylem and the phloem. And so jasmonic acid or methyl jasmonate. And then GLVs, I think we've mentioned that, or it will be in that video. That means green leafy volatiles, GLVs. It's just kind of an umbrella term for a bunch of different volatiles that plants give off, usually to defend themselves. So that made people start thinking, okay, why are they, I mean, they have choices here. Why don't they just do it through their own body? Like, why would they give off the volatile? I mean, for the parasitoid wasp, that's one thing, but this whole idea of neighboring plants, if you think about it from the idea of plants compete with each other all the time, and which, they are competing for sunlight. So you might think, well, why would they give off a volatile that they're getting attacked to their neighbors? Because if their neighbors get attacked and die, then they'll have more access to sunlight. So that must mean the neighbor plants are just eavesdropping, and the plant really didn't intend to give off that, hey, I'm getting attacked signal. That makes sense? And so that was like one of the first thoughts, like, that doesn't really make sense that they give off a volatile. But I think that's a pretty simplistic way of thinking about it, because neighboring plants don't always just compete with each other. there are a lot of interactions going on that, taken as a whole, that little ecosystem can really benefit from having neighbors, even neighbors of the, same species, different species. And I think that initial thought of, well, they're always in competition, that's a little too simplistic. And we're going to look at, when we start looking at plant-plant interactions and forested ecosystems in particular, that it makes sense that you do have neighbors and that you have interactions going on even between different species that can be very healthy for the entire ecosystem. And so I think that kind of falls apart a little bit, this idea of they just want to kill each other so they can get more access to sun. That's kind of a holdover from decades ago. And basically, too, I mean, the idea of Charles Darwin and natural selection, right? Only the strongest survive, survival of the fittest. And so it's kind of a holdover on that idea, but I think they're putting too much emphasis on it. So here, a couple of things. So here they're saying it again, what I was just trying to get across, this idea of competing for light. So why would they put out a volatile to tell their neighbors to gear up? And then they say one of the reasons could be, and there's this word, orthostitchy. Don't get too caught up in that. Basically, what that means is how directly connected are the plant parts to each other. So if a beetle is chewing on one leaf, then a leaf on the other side of the plant, it's not a very direct connection. And so you would say that it has high orthostitchy. Just think about connections. I think they put it in here a little bit further down. Directly adjacent leaves usually lack direct vascular connections. And so this idea of adjacent leaves, they don't always have a nice connection with their xylem and phloem, the vascular connections. And then if you go on to the tree idea, that they have a lot of indirect vascular connections through the whole plant body. And so if one part of the plant's getting attacked, it's going to take a long time to put that signal through your vascular system and go entirely internal to the other parts of the plant that don't have very good direct internal highway systems, essentially, because the vascular system, you can kind of think of it as an internal highway system to get chemicals moving through. So, and then they get into a little bit more detail here of how things move through the phloem and how things move through the xylem. Basically, they're saying that just the physics of how things move through those two parts of the vascular system, you can't always get a signal to distant parts of the plant. And so from that perspective, it does make sense that there's a volatile. So even that one plant that was getting attacked by that beetle, the fact that it was giving off a volatile, if you think about it, especially a huge tree, then the whole canopy theoretically could get exposed to that volatile pretty quickly, compared to if you just try to go through the internal highway system of the plant body. And that can be very slow, and sometimes it just doesn't work. You can't easily get things from point A to point B if there's no direct connections. Okay, so that's kind of the thinking right now, that volatiles are necessary even for the same plant, especially if it's a big plant, that volatiles might be a pretty necessary thing. Still doesn't get at the idea of, is this eavesdropping? Is it done intentionally? We'll come back to that idea as we work through. Once we get into the plant-plant interactions, we'll start thinking about that a little bit more again. Have I convinced you VOCs can be important? Okay. Whether or not the plant's being nice to its neighbors and saying, hey, gear up because I'm under attack. From what I've read, my personal conclusions on everything I've been reading, I think you have to take it on a case-by-case basis. But so much of what I've read, I'm leaning toward the idea that this is done intentionally by the plant. But I'll try to convince you of some of those papers little bit later when we get into the plant, some of the research on that. So I just included a couple quotes here by Monica Gagliano, who did the dropping of the mimosa over and over. And she said, if we think we already know how things are and fail to continuously question our own assumptions, but construct our claims on a system of beliefs we are dearly attached to, then we are in deep trouble and miss the opportunity for true scientific discovery to occur. So this is a quote that she gave to a interviewer because her work has, and she herself, she has been attacked in the journal articles, people writing rebuttals and at conferences. Have you ever gone to a big scientific conference? People can get nasty. I mean, someone will be giving a talk on stage and someone in the audience will like get up and shout at them. I mean, it's just like, really? So it can get nasty. And so this is some of her kind of pushback on this. And and she's not the only one. There are many scientists, I've read them being interviewed, that are in this field of signaling and behavior of plants. And so many kind of echo this idea that you can't just hold on to your favorite idea. Yeah, with new ideas, you have to keep an open mind. Yes, do experiments, get the data, but don't immediately knee-jerk say, no, that's ridiculous, especially if you have no data to back that up. You need to be able to show some data that if you're disagreeing about something. And so the other thing too, oh, one more. It's to me, the role of science is to explore and to explore especially what we don't know. But the reality is that much research in academia tends to explore what we already know because it's safe. And that is very true. After being where I am now at my career stage, I see this ringing true, unfortunately. And I think part of the reason is grant funding, because granting agents want to play it safe. Because if they're going to give you millions of dollars or hundreds of thousands of dollars, they want you to do something that's going to work. I mean, you can kind of get it from their perspective, but scientists can't do groundbreaking research without good funding. So it's this chicken and egg kind of thing where it's really hard to make progress if you need a lot of money to do that experiment. And so this is something that I don't know what the answer is for that. Now with grant funding getting cut even more severely, because politics play into this, of course. And so and there's a time when, unfortunately, any experiments that had anything to do with learning or memory or senses were just totally like, nope, we're not funding any of it. And it's actually due to a book that was written back in the 70s by Peter Tompkins and was published. And it was a lot of pseudoscience. And so the scientists were up in arms because it really wasn't a very good book. There's a couple sections that were worth reading, but it was all about plants, prefer classical music over Led Zeppelin and, this kind of thing. And just no good science to back it up. But it kind of caught on. People that were not scientists liked that idea and ran with it. So it became very, very popular. But the scientists were like, oh, no, don't have any data to show any of this. That's really good data, no sound experimental design. And so the granting agencies just kind of put a total, like, what's the word I'm looking for? They just said, we're not going to fund anything like that. And so for decades, nothing on plant behavior was looked at because you couldn't get funding for it. And not only that, but if you tried to do something, you'd get harassed by your fellow scientists. So it can get kind of vicious. It's a soap opera world out there sometimes when it comes. I'm sorry to say, if you're going to go on into academia, it's not for the faint of heart. So be careful and know you're not alone. This is kind of unfortunately the way it is. So I'm hoping it'll change. I'm hoping the next generation will enlighten people better. All right. Questions on that? Any of those ideas? plant signaling and behavior.
Have they ever related it back to trees in a way, because trees work in like a community like interaction with like how they kind of support each other, like if you cut down the tree. They're still like their root systems will be connected and they'll keep each other alive by giving each other nutrients. Would it be necessarily kind of correlated to that? Like
Yep.
they're involved in like those similar
Yeah,
traits?
exactly. So, and we're actually going to get into that a fair bit with plant-plant communication and the mycorrhizal fungi that also are serving that facilitating role. And so, yeah, I mean, it's some of those ideas, especially in a mixed forest system. There's been some really nice studies showing that there is communication going on and that it is done on purpose, and to help that neighboring plant. And so we'll get into that when we do the plant-plant interaction. I think, I can't remember which class I mentioned this in, but one of my neighbors cut down a beautiful oak tree that had to be, for sure, I would say at least 100 years old, if not closer to 200. It was massive. And they just bought that house and they just moved in. So they were new to the neighborhood. And I was walking home one day and saw the tree cutting business cutting it down. And I went over to the tree cutting business guy. I said, why are you doing this? Because the tree was healthy. And he said, the owners want it cut down. And I said, why? Reason did they give you? And I said, it's not diseased. And he said, they didn't want to rake the leaves. And to this day, I can't talk to those people. I just can't. I just, and the guy who was doing it, he's like, it's my business. I have to do it. He says, I'm heartbroken to do it. He was like really bumming out that he had to do it. So I gave him credit for feeling bad about it. But yeah, and so that mature, huge tree, it wasn't just that new owner, his yard that was being impacted. It was all the trees in the neighbors now because those roots were connected to the neighboring trees. And so it has this trickle down impact on the neighbors and their trees. And there's a patch of woods across the road. So all those trees, there's probably all those it's lost now. And so it makes me wonder what those other trees are feeling, so to speak, with the loss of that. I mean, that was like I would have considered it like the mother tree for that neighborhood, and it's gone. And I see that so often. I talk to somebody else, a friend that has a patch of woods, and he's like, yeah, my friend wants to cut down all the big trees in my forest. Like, why? Well, that'll give them more money, right? The bigger the tree, the more money you get for it. And I said, but you are now creating a situation where the remaining part of the forest is going to be at risk. And it was really hard to get that point across because he's not a scientist and certainly doesn't really know anything about plants. And so it's very frustrating because the knowledge isn't out there, and common knowledge, but hopefully I'm hoping that you'll all be advocating as you learn more through this class, that some of the knee-jerk ideas that people have about plants and forests is not correct. So, but anyways, all right, let's take a break. Come back at 3 and we'll get into the next slide set. Okay, so epigenetics. Some of in the plant world, often you'll hear or see something published that says plant transgenerational memory. It's basically the same thing as epigenetics, but just be aware that they had to get fancy with their terminology. So here we're looking at, and at least from the readings I've done, epigenetics was first discovered in plants. They didn't discover it first in animals. That's at least what I read. So this was a more recent paper. This wasn't the very first. This is back in 2005, but this is, within recent history, this was one of the landmark papers when it comes to epigenetics. And so Plants rewrite their genetic code. So this, I like the start of it, in a discovery that has flabbergasted geneticists. They have shown that plants can overwrite the genetic code they inherit from their parents and revert to that of their grandparents. This challenges textbook rules of inheritance. And so this was published in in the journal Nature, which is like one of the journals. If you publish there, that's a huge gold star for you, and you're more likely to get funded. It's a big deal. And so let's get into this a little bit. This was actually the title, the paper that they were referring to, and you're not going to read it because it's complicated, but this is the start of it with a little intro or abstract. And so this is at Purdue University. And so, of course, they gave it a big long title that's hard to make sense of. Genome-wide non-Mendelian inheritance. So that what they're saying is it's not due to a mutation event of any of the bases of the DNA. Extra genomic information, so like methyl groups or acetyl groups, all right? And Arabidopsis is the guinea pig plant that is commonly used. So in that video, you heard some definitions of what that, what does epigenetics mean? So this slide is different ways of stating it. Whatever works for your brain, go with it. It's all basically saying the same thing. So, you really are changing the gene activity, not any kind of mutation event with the DNA bases. All right. It's whether or not the genes are turned on or off. And you can do that through this epigenetic mechanism. And it can get passed into offspring. And so it changes the structure of the DNA. So what they mean by that is it could relax pieces of the DNA. They gave that example in the video where it was wrapped around the histone proteins real tight. And if you can relax that, then it's more open for the RNA polymerase to come in, latch on, and start transcribing that section of the DNA to make some proteins. And so relaxed DNA, better access, tightly wound up DNA, RNA polymerase has a really hard time getting in there, and so the gene will be turned off. And so you are kind of looking at what's getting turned on and off, not what's mutating, right? Defined as the study of heritable trait changes of DNA, not involving changes in a DNA sequence that regulate gene expression. And this very last point is what I tend to memorize: a change in phenotype without a change in genotype. To me, that makes sense. And so that's kind of like the quick and easy definition of what epigenetics is. It's accurate. Okay, so the idea that plants could do this, like I said, from what I read, plants were the first organism to have this discovery. And so this is just getting at a little bit of what DNA looks like. So if you need a refresher on it, it's a double helix, it's wrapped around histone proteins, it's very tightly wound, incredibly tightly wound. I like that, overly twisted rubber band. That's a good analogy. And so active genes are found in areas where things are unraveled or loose, while inactive genes lie in more condensed regions. And that just has to do with the fact that the RNA polymerase needs some physical space to get in there and actually attach onto the DNA. So a visual mock-up of what we were just talking about. It's pretty incredible when you think about it. It's mind-blowing. And so here, it's not specific to plants. Paradigm shift, definitely. This has really changed the field of genetics absolutely. Epigenetics, it facilitates memory, not only from season to season within, with plants, within a single organism, but from generation to generation. So one of the papers that was published in the early 2000s was done by Dr. Barbara Hone, and she was looking at plant stress, kind of, like they were looking at those poor mice that weren't getting any mothering versus plants or mice that were getting well taken care of by the mother rat or mouse. So in this case, we're looking at stressed plants And she was able to tease apart that it's not a mutation going on, but that you have different gene expression going on. And she could track that the offspring from that mother plant had that same altered DNA activity, even if they weren't exposed to that stress. All right. So the seeds that the mother plant made, it's not an active plant, it's a seed. And so you find in that seed that it has the changes, but it never directly experienced when it was, growing. So the stress in the parents caused a stable, heritable change that was passed on to all the offspring. The offspring plants behaved as if they had been stressed when they grew up those plants after they germinated the seeds. So they remembered that their parents had been through this stress and reacted similarly. So this was, like I said, back in 2006. It was one of the more current studies that were done with some good experimental design. It was done in plants, not animals. And she was getting some pushback, but this is taken from someone who was supporting the research. The use of the word remembered may be unorthodox, but if you think about it, the parents formed the memory of the stress, retained it, and passed it on to the children. And the children recalled the information and reacted accordingly, in this case with increased genomic change to deal with stress. And so you can get this heritable change going on into successive generations, which again has a little bit of the feel of Lamarckism. And so again, it's interesting to see how, at least when I was an undergrad, people would just be so dismissive and kind of nasty about Lamarck's ideas. And now it's coming back again that, oh, but wait, there might be something to this, at least in some cases. There might be something to it. All right. So she was doing, you don't really need to, I'm not going to post her study because it's intense, but basically, if the stress induced an epigenetic change, this could happen in all the cells at once, not just pollen and egg, but all the cells, all the body cells, pollen and egg cells as well, and it can get passed to the next generation. And so they still are trying to tease apart how this is exactly happening, but basically, this is her study, the one that I've been talking about, transgeneration memory of stress in plants. And so this was looking at, they took it out to 3 generations, and they could show that the first and second generation behaved as if they were experiencing the same stress as the mother plant. They had all the gene activity to deal with it. But then by the 3rd generation, that went away. And so they reverted back. And so this is something, so there's this plasticity to it. It doesn't mean it's permanent. That's why, they're saying if you eat well and don't get exposed to a lot of toxins, you can change your gene expression and get a healthier response of your body. So when that was published, there was pushback, surprise, surprise. And so there was a paper written by Pachinka et al. Transgenerational stress memory is not a general response in Arabidopsis. This was published in 2009, so 3 years later. And which I didn't post this one, but basically this group of scientists were like, no, that's wrong. But then another paper published in 2009 and others beyond that started to show more evidence that was showing that epigenetics is really happening in plants and it can go into the next generation. And so here, looking at this idea, I didn't post this one, you don't need to read it. And then another one in 2011, this one was done with fruit flies, I believe. And looking at how it's inherited. And so that was some really good data. We're not going to get into the actual experiment, but it was, a good piece of data that was being added. Another 2011 paper. This one was done in plants. This one was really interesting because they looked at trichome numbers on the leaf. And as you know, sometimes trichomes carry some very interesting secondary metabolites. So this information could be applied to the idea that if you want plants to express higher numbers or certain types of secondary metabolites in those trichomes, this kind of idea could be something that you would play around with. But this idea that a transcription factor, so that's the transcription factor that latches onto the DNA and says, here, transcribe this gene. And so they could show transgenerational inheritance, and it had to do with trichome density in this particular plant. And so it wasn't due to the DNA sequence, but to the chromatin modifications. So basically they're looking at the idea of maybe it was methylated or acetylated. And so in this particular case, it increased the trichome density in response to simulated herbivore, and then it went into the offspring. And so seeds from that plant would have more trichomes. And so if you're interested in something that comes in trichomes, this could be something to think about. And so the idea of, this epigenetic transmission into the next generation. And so this was back in 2011. And then they came up with another set of data in 2014, where they were looking at dehydration, so drought, simulating drought, and they were able to show that the memory of the stress response. In this case, they did it with this. I don't think this was transgenerational. This was more from the plant first experiencing drought and then recover it and then make it go through drought again. It did better the second time around. So this wasn't transgenerational, but it was still showing this idea of epigenetic modification of the DNA. In this case, it was, well, it looks like they're not sure yet. But they mentioned histone modifications and DNA methylation. And then there's been some really recent work being done. 2024 epigenetic regulation. So this is a review paper. So I put this in here to demonstrate that if you're able to write a review, that usually means you have reviewed and assessed and gone through a lot of papers on this subject. So the fact that they're able to write a review paper about epigenetics, they looked at 401 studies in order to write this review. So I put this in here just to emphasize they've come a long way since the early 2000s in terms of research on epigenetics. And the data is definitely accumulating. And then this one in 2025, looking at, this was really cool, looking at epigenetics in the interplay between beneficial like PGPRs and plants. And so now they're even considering that role as they think about how plants and their beneficial microbes interact with each other. So the data is just growing from about 2005 onward. Initially, there's, pushback, but now, over 400 papers and far more than that have been published on this idea. So this is something that's still, even with hundreds of papers, there's a lot of work to go on this yet. But basically, what they're finding in the plant world is that it seems to be even more important than in the animal world, which makes sense when you think about how plants have to just deal with their surroundings. They can't get up and move away from threat or stress. And the fact that they can have this plasticity to their gene expression and then pass that on to their offspring. So their offspring have a chance, if this environment doesn't change, the mother plant's going through a stress of drought, and then they pass on the ability to deal with that to the offspring. If they germinate and grow in drought, they'll be able to survive. So it makes sense that plants actually have a better handle on this than animals and do more of it. So when you look at, I'm just going to say a little bit about how it works. Basically, they're seeing some interesting ideas. That video talked about methyl groups being tacked on to the DNA, or acetyl groups, and the histone proteins getting modified also along with that. And then they didn't mention the non-coding RNA. I'll mention a little bit about that. But again, that you start to get into the weeds on some of this stuff. But basically that middle pink oval, they're saying that all of these are influencing gene activation. And then, of course, I guess I would just say gene expression. I mean, they say gene activation or silencing. So they're saying, you know, turning on or off. Gene expression in general is altered. And of course, that's going to have trickle down effects with what happens to those cells and tissues as well. So methylation, this is just a mock-up trying to show the methyl group. And it does seem to always stick on to cytosine. That's what they're finding. You know, there's 3 other bases in DNA, but it seems like it's always the cytosine that gets tagged. And so they're showing in the lower portion how if you have methyl groups, these black balls down here, it tends to close things up or condense the DNA. And then that stretch of DNA doesn't get expressed in this case. And then the unmethylated keeps things loose and open so that that section can get expressed. I put this down here just in case you've forgotten about your genetics class. That's just basic idea of what a gene looks like. I'm not going to ask about it. It's just for your purposes. Histone modifications. So they talked about that in the video. They have tails, so to speak, of amino acids. That's what they're trying to show here with the warm white purple line. And so you don't need to really know that, but basically this idea that you have the modification going on the histones, specifically the tails, they've been able to figure that out. And so they're showing, again, if it's condensed, the DNA is tightly wound around the histones, no gene expression. If it's relaxed, then you can get the transcription factors in there, physically get in there and latch on. And then that allows the RNA polymerase to get in and do its job. So the acetyl groups, I would like you to at least know that acetyl groups will relax. In general, that's what they're finding. Acetyl groups do relax. Methyl groups tend to make it tight. All right, so the idea here, at least in plants, what they're finding so far is that it's definitely playing a role in the process of flowering, if you're an angiosperm, seed development, and a lot of abiotic factors have been studied, but even biotic, like fungal attack or pathogen attack of some sort. So you can see, I mean, all of these are super important to a plant, you know, being able to flower at the right time, in the right way. If you don't do that correctly, you're not going to get anything going on into the next generation. You have to have flower production, pollen and egg production. Seed development, obviously super important to get your genes into the next generation. You have to have good seed development and have that happen at the right time of the year. I mean, all of this is so important to the plant that you can imagine that there might be all of these epigenetic controls to make sure everything goes okay. This is just nothing I'm going to directly in detail ask you about. It's more that if you've not thought about RNA in a while, most of us are pretty familiar with messenger RNA, transfer RNA, and ribosomal RNA, but if you've not taken BI-218 or advanced cell, there's a bunch of other types of RNA out there that they have discovered. And non-coding ncRNA is one that within the non-coding bracket. It's not even on here because it's relatively new. This paper was older that I grabbed this image from. They've come up with long, do I have, whoops, long non-coding RNA. All right, so these are longer stretches than what they initially found, so they gave it a novel name, LNC RNA, long non-coding RNA. And so they've narrowed it down to that one is causing flowering in some of the studies that have been done. They've even named the gene. Cool air, you don't need to know that, but if you mess with it and get rid of it, the plants won't flower. That's how substantial the impact is. And so this idea that the timing of flowering and the process of flowering. And so not only do we have to think about methyl groups and acetyl groups, but now these non-coding RNAs, long non-coding RNAs seem to be involved in this epigenetic regulation. And so they're probably going to find more out there as they get along. Like I said, there's lots of different RNAs out there, so those other ones might be involved, but at least that one study on flowering, they could pinpoint the LNC RNA. So altogether, at least so far, what I'm finding in the literature is that flowering is controlled through epigenetic mechanisms. So is cold adaptation, drought stress, salinity stress, biotic stress. They've looked at like herbivory or like fungal pathogens primarily, I think. I don't know if they've done much with bacteria yet. So that's the idea of biotic stress. Whereas cold, drought, salinity, that's mostly abiotic ideas. And so with the biotic stress, they're looking at how does the plant respond to attack in terms of its immune system? How does that get regulated? And then how does it form a memory, so to speak, so that when it gets attacked again by the same pathogen or herbivore, it will be able to respond that much more quickly. So the research is going on still. And my hat's off to people who do this kind of research because it's not straightforward. The deeper they get into it, it's complicated. And that's as deep as I'm going on this topic because this is not a genetics or molecular biology class. But I do want you to think about it in the sense that it really is kind of a nifty way for a plant to have a very plastic way of dealing with its environment at the genetic level, and then getting that into the next generation so that their offspring hopefully will survive if they're met with a similar fate of whatever environment the mother plant might be in. Questions on epigenetics? Kind of makes sense. When I was first learning it, I had to go over it a couple times. So if you're like me, make sure you give this some thought outside of class. Rewatching that video might help as well. But that's it for the exam material. No more for exam 1. I have not finished making the exam, so I don't have anything yet to say on length. But when I know more, I'll let you know. But essentially, it's next Wednesday in here, EduCat, mostly essay, and the whole class period. Okay? So today's Wednesday, right? So we'll meet on Monday. By Monday, I'll have a better feel for the exam, and hopefully part of Monday we can leave open in case you have questions and want to revisit some topics. So I'll probably leave the second half of the class period for that. So we're all together and can share questions and answers, okay? All righty. Why don't you go out there and sweat in the sunny weather?
A more specific list.
Yeah.
Is that